everyone. My name is Kaoru Sakabe. I am the Data Integrity Manager for ASBMB. In January of 2019, I was invited by Johns Hopkins Medical Institutes to give a responsible conduct of research seminar on transparency and reproducibility in publishing. It just gave a very broad overview of some common issues that authors may face while they're writing up their research results, as well as some consequences that may happen if authors don't follow best practices. We are very fortunate that Johns Hopkins shared a copy of that recording with us, so I hope you enjoy the recording, and thank you very much for your time. So, hello everyone. Thank you all for coming today. I'd also like to thank Karen and Melissa for inviting me here to speak to you all. It's a real honor to be here because I am an alumnus. I never would have thought I would be giving an RCR talk in the same auditorium where I saw it many, many years ago through RCR lectures myself, although the seats were much more uncomfortable and there were chalkboards up here. So things have changed. So as Melissa mentioned, I'm the Data Integrity Manager for the American Society for Biochemistry and Molecular Biology. So we publish 3 journals, the Journal of Biological Chemistry, Molecular and Cellular Proteomics, and the Journal of Lipid Research. So the JBC actually has a very long history that's intimately tied with the Department of Biological Chemistry here. John Jacob Abel, who was the founder of both ASBMB and JBC, actually founded the Department of Biological Chemistry. So it's actually a real double honor for me to be speaking to all today. So one of my main responsibilities for the Society is to function as their publication ethics manager. We want to ensure that we handle any issues that arise in our publications fairly as well as consistently. And now, because we are a society, another one of our major goals is to make sure that we are educating our potential authors as well as the scientific community as a whole on best practices in publishing. So that's what I'm going to be talking to you about today. If I can get the slide to go forward. So again, here's just a brief overview of the topics I'm going to be covering today. First, I'm going to walk you through some best practices during the experimentation process, as well as manuscript preparation. And then the very brief second section of my talk, I'm going to be talking about some consequences that can happen to you if you don't follow these best practices. So you guys are all researchers. You're probably all very familiar with this research life cycle. So I'm not going to walk you through all the steps here, but I'm just throwing this up just so you can keep in mind that science really works by building upon prior knowledge. We wouldn't be where we are today if we didn't trust previous work, and science is not going to advance any further unless we continue to perform rigorous and transparent research. So in other words, science needs to be conducted with integrity. Otherwise, how are we going to be able to trust what we're reading in the papers, and how is that going to further our current knowledge? So in fact, the National Academies recognized and wanted to address this topic. So in 2017, they published a report on fostering research integrity. So while the report did acknowledge that research misconduct is harmful to the research enterprise, it also acknowledged that detrimental research practices are just as compromising to research as misconduct. So these detrimental research practices include behaviors that lead to irreproducible research. So my talk is going to focus on how you can avoid these through some best practices. So some of the things I may go over may seem really tedious or like unnecessary busy work, but remember, these are best practices that are meant to safeguard your work as well as foster integrity. So another small apology and disclaimer before I get any further. I know our audience is a bit mixed with both basic scientists and clinicians. So my background is in the basic sciences. So a lot of my examples are going to draw from there. But I think a lot of the concepts I'm going to be talking about to you today are going to be applicable to everybody here. So again, here's just a brief overview of what I'm going to be talking about. The first thing I'm going to talk to you about is about experimentation and data analysis and collection. So when you start off with a research project, you're going to start by asking a question. How does protein X associate with DNA? Are you going to improve patient outcome by inhibiting protein Y? So you start with a question, you come up with a hypothesis, and then you're going to test that hypothesis by doing a bunch of experiments. In order to test that hypothesis, you're also going to need an experimental system. Now, this can be a cell line, a purified protein, a mouse line, or even human subjects. So you perform experiments in your system, And then for you to draw conclusions from those experiments, you need to perform independent repeats until you're sure what you're seeing is an actual effect. So a point of confusion I see among some authors is the difference between biological and technical replicates. So biological replicates are biologically distinct or independent samples, and tells you something about the natural biological variation of whatever experimental system you're looking at. Technical replicates, on the other hand, take measurements from the same sample and only shows you noise associated with your equipment or particular experimental protocol. So let's take, for example, you're looking at the proliferation of cells from a bone marrow suspension from 2 different genotypes of mice. So you take 1 mouse from each genotype and prepare 3 plates from a single bone marrow suspension. Now these 3 plates are going to represent 3 technical replicates and are only going to tell you errors associated within the assay itself and not the true biological variation of your experiment. So here, in this case, n is going to equal 1. It doesn't matter if you're going to measure 3 plates or 100 plates from each mouse. N here is always going to equal 1. On the other hand, let's say you decide to take 3 different mice from each genotype and prepare 3 plates from those mice. So in this instance, you're now measuring 3 biological replicates. So n here is going to equal 3. Again, it doesn't matter if you're measuring 3 plates or 100 plates. N here is going to equal 3. So this information, that is the number of biological or technical replicates that you've done for your experiments, is really important to note because it gives readers a sense of the quality and the reproducibility of your work. So also, as you're designing your experiments, you're probably going to be ordering reagents such as cell lines, chemicals, and antibodies. Now, how sure are you guys that you're working with what you think you're working with? Just because you've gotten the resource from a close collaborator or a commercial entity doesn't mean you can trust that resource. So for example, the Chang liver cells originated in the early 1950s from a patient biopsy. And then it was deposited into the American type culture collection in 1962 as a normal in vitro liver model. Between about 1967 through the 70s, there were several papers that were published that argue that these liver cells are actually a contaminant, HeLa cells, which is a cervical cancer cell line. In 2001, definitive proof was published that established that the Chang liver cells are in fact HeLa cells. So even with all the evidence that's been published, some over 40 years ago, there are still papers being published today that are using these cells as a liver model. In a more recent example, this article that was published in Nature Cell Biology in 2012 was retracted by the authors in 2015. Now, if you look at the notice, the authors state that the antibodies, or some of the key antibodies that were used in the study, were nonspecific. And then if you go look at the materials and methods section, you can see that all the antibodies were purchased from commercial suppliers. So I would say having your paper retracted is a pretty hard way of figuring out that your antibodies are nonspecific. So I would strongly encourage you all to validate your reagents before you even start your experiments. So besides your reagents, when you're planning your experiments, you should think about whether or not you're running the appropriate controls. Now, controls are going to allow you to eliminate varying amounts of uncertainty in your experiments. So I'm not going to go through the different types of controls that are listed here, but before you start your experiments, you should think about your experimental system that you're looking at, and then what controls you're going to need to verify that your experimental system is robust and to eliminate alternate explanations of any of the experiments you've run. So one control that is extensively used when running Western blots are loading controls. So loading controls are used to determine if differences seen in protein expression levels are due to actual differences in your experimental conditions or due to differences that may happen during electrophoresis or transfer. So what most people will do is they will probe for their protein of interest and then just make sure everything is legit by probing for a housekeeping protein such as tubulin or actin. So the big issue here is that these housekeeping proteins that are normally used as loading controls may not actually assess equal load. So in this paper that was published in PLOS One, what the authors did was they took 15 micrograms of spinal cord extract, either from a wild-type mouse or a mouse model of spinal muscular atrophy. And they ran them out on 2 different gels. So one gel they stained for total protein here in red, And then the other gel they transferred and probed for 2 different housekeeping proteins, either actin or tubulin here in green. So when the authors quantitated the change in relative expression to wild type, they saw that both actin and tubulin were downregulated in their mouse model, whereas the total protein stain was uniform. So in fact, when the authors went on and looked at several different commonly used loading controls, they saw that they were not equally expressed in several different mouse models. So finally, the authors go on to demonstrate that the signal detected from commonly used loading controls, such as actin or tubulin, saturates or is no longer in the linear range at about 10 micrograms of protein. So most authors or researchers are loading much more than 10 micrograms of protein when they're trying to detect their protein of interest. So this means that these common loading controls that people use are actually not functioning as a real loading control. It's much better to look at looking at total protein uses stain, because this has a much broader linear range. So if you do have to use a housekeeping protein, like if it's an absolute must, what I would recommend demonstrating to yourself is that not only are you able to detect it in the linear range at whatever protein amount you're loading, but also that its expression level doesn't change with whatever experimental treatment you're using. So here's another example. Here the authors have treated their cells with either drug X or drug Y for 6, 12, and 24 hours. So what they wanted to look at was to see if phosphorylation of proteins A, B, and C increased during this time course. So they probed with antibodies that detect the phosphorylated species of these proteins. Do you guys have any idea what the authors may have done wrong here? Does anybody want to wager a guess? You guys want the answer? Okay.
I was thinking while you're doing this, you fix this.
Oh, okay. So the first issue is that the authors normalize the phosphospecies to actin, but they don't actually demonstrate that the total protein abundancy of these proteins aren't affected by the drug treatments. So ideally what the authors should have done was probe for phosphoprotein A and total protein A, phosphoprotein B and total protein B, et cetera, et cetera. So the other thing is we're not really sure if actin is affected by either of the drug treatments the authors used. It would have been better to look at total protein by using either Kumasi or Ponso-S. And finally, if you just look at this actin blot down here, you see that it's completely overexposed. There have been no way for them to determine if they actually loaded equal amounts of sample in the blot based on how overexposed this blot is. So the final thing I wanted to mention about your experimental design is you want to make sure that you're comparing equivalent samples. You want to be sure you're able to draw conclusions from the experiment that you're running. So for example, let's say you're treating HeLa cells with a drug and you want to see how it affects a certain protein. You wouldn't be using PC12 cells as a negative control. You're going to be using HeLa cells that were treated with vehicle. So building on that example, so here is an experiment where the authors wanted to determine the binding of these 3 GST tag proteins to either full-length protein X or its N or C terminus. So what they did was they immobilized either MBP or MBP tagged versions of either the full-length protein N or C terminus 2 beads. And then they performed binding reactions by incubating these beads with these 3 GST tagged proteins. So then they washed them a few times and ran the bound proteins out on gel, transferred them, and probed them for either GST or MBP. So when we first took a look at it, the thing that really struck out to us was the fact that there were splices between each and every one of these bands here. And then we also noticed that these MBP tagged proteins are presented to make it look as if they were all migrating at the same molecular weight when they should all be different sizes. So when we dug a little bit deeper with the authors, we found out that they had run a lot of these assays out on different gels. And they didn't use a standard in between those gels to make sure they were accounting for differences in transfer efficiency or exposure time. And then finally, they were doing quantitation on these blots, on completely overexposed blots, so they wouldn't be able to accurately quantitate the amount of signal that was in each band. So talk to you a little bit about your experimentation, but I'm going to go over a little bit about your data analysis and collection. So I am going to preface this by saying I am not a statistician at all, but these are just a few basics that I think everybody should know. So first, I think we often pressure ourselves to demonstrate that our results are a lot more significant than they actually are. So we put error bars on quantitative data when in fact it's not appropriate to include them. So in A, what the authors have done is they've taken one culture of cells and treated with medium, and then the other culture of cells they treated with the cytokine HHCSF. So they extract RNA from these cells and then quantitate the ratio of Boojum mRNA to actin mRNA using quantitative reverse transcription PCR. So these three squares here represent 3 aliquots taken from the same starting material of RNA. So in this situation, it's not appropriate to include error bars because these represent 3 technical replicates and only tells you that the pipetting was performed consistently or the PCR wells were heated evenly. Now, if the authors had taken 3 independent cultures of cells, here, here, and here, and treated with or without cytokine and measured the mRNA in triplicate, these are 3 biological replicates. So in this instance, the authors would be able to use error bars. So next, are you displaying your error bars correctly? That is, do you know the difference between standard deviation and standard error of the mean? So standard deviation quantifies the scatter or how much the values vary from one another, while standard error of the mean quantifies how precisely you know the true mean of the population. Standard error of the mean is always going to be smaller than the standard deviation, and it's going to get smaller the more samples that you have. So if you want to show the true variability or the true spread of your data, you really need to be plotting your data showing the mean plus or minus the SD. Now that said, there is a push coming from within the scientific community for authors to be more transparent in how they're showing their data sets. So for example, this figure shows the same data set plotted 6 different ways. So this plot here, which is the scatter plot, it shows all the individual data points that you obtained in your experiment. The box and whiskers plot shows the whole range of the data, the quartiles, and the median. Next is the median and the quartiles. And the next 3 are just the mean plus or minus the standard deviation, the confidence interval, and the standard error of the mean. So while it's generally acceptable for you guys to plot the mean plus or minus the standard deviation, I would highly encourage you guys to plot your data using either a scatter plot or box and whiskers because it's much more informative for readers to see the entire spread of your data. So here's another example of why plotting all your data is much more informative to readers. So here, if you were to take all of these plots shown in B, C, D, and E and show them just as mean plus or minus the standard deviation, it's all going to look like it does in A here. But if you look at the raw data, you can see that there's actually a lot of good information that's going to get hidden if you show it just as a bar graph. So in C, what happened was an outlier caused the data to skew to make it look like there was an actual difference between these 2 experimental conditions. In D, the data are distributed bimodally. And in E, there were an uneven number of samples between the 2 conditions. So if you must absolutely plot your data using a bar graph, what I would suggest or recommend is to superimpose these individual data points on top of the bar graph so that the readers have an idea of what the spread of the data are. Otherwise, like I said before, I would highly recommend plotting all your data using either a scatter plot or a box and whiskers plot. So the other type of data you're going to be collecting while you're doing your experiments is going to be image data. So a mistake that I see quite often in images collected by authors is oversaturation or the need to eliminate any background noise that's associated with that image. Just remember, there's intrinsically noise in any type of biological system you're going to study, so actually having that background in your image is a hallmark of true biological data. So one way you can demonstrate to yourself that you've acquired your image appropriately is just by looking at the histogram or the distribution of pixel intensities in your image. So here is an image of the cytoskeleton of a mollusk embryo and then its corresponding histogram. You can see that all the pixels fall within the range of intensities and nothing in it is purely black, which is here at 0, or purely white, which is here at 255. So the image next to it here has been appropriately processed. If you look at the histogram, you see that none of the pixels are purely black or white here. And the other thing that's really important to note is that the shape of this histogram hasn't changed from the original. That means the information that was contained in the original image has not changed. So now if you look at this third image, this one has been aggressively manipulated. These spikes here at the black end and the white end mean that these pixels are saturated. And then if you zoom into these boxed areas here, you can see that a lot of that fine detail that was in your original image here has disappeared once you've made that adjustment. Another thing, if at any point you need to quantitate your data, you're going to be underestimating the amount of signal that was in your image if it's saturated, and you won't be able to tell by how much. So here is kind of a ridiculous example, but we're going to look at this overexposed blot here. If you look at the histogram, you can see that the most abundant pixel in this image is 0 or pure black. And if you then attempt to perform densitometry on this upper band, something I would definitely not recommend, you can see there's hardly any variation in the amount of signal that you're able to detect. Now, if you take a more appropriately acquired image, such as this blot here, and perform that same calculation, you can see that there's now variation in the amount of signal you're detecting. So I know you're thinking about making sure you've acquired your data appropriately, but you also need to make sure you're saving your data in a format that's going to be usable down the road once you're ready to submit your paper to a journal. The bare minimum, that means you need to be saving in a universal file format that uses a lossless compression at a minimum resolution of 300 dpi. That means you should be saving your images as TIFFs and not JPEGs. If you've used an imaging system at all, I would also recommend retaining a copy of that data in native format, just in case if you need to go back to it later to re-export an image. So JPEGs are a huge issue when it comes to publication quality. And the reason is that the algorithm used by JPEG compression fundamentally alters your data. And it does this by throwing away some information that was contained in your original image. So here is an image of the word JPEG that I've saved either as a TIFF or various quality JPEGs. Now, if you look at the histogram of the image that was saved as a TIFF, you can see that most of the pixels are either white or black, and that there are some shades of gray here. Now, as you save it in progressively lower quality JPEG saves and look at the histogram, you see that pixels are getting introduced into your image that didn't exist in the original here. So how is this going to affect your scientific data? So here is an image that I've saved either as a TIFF or, again, various quality JPEGs. I then took these images, put them into ImageJ, and did a surface plot analysis on them. So here, even with this high-quality JPEG save, you can see that the image already looks different than the original capture here. And then when you get to this low-quality JPEG save, you can see that the bands look really pixelated and blocky. So not only does one save in JPEG alter your data, but each time you're saving that image as a JPEG, that compression is going to get reapplied. So here's an image that I've saved either 10 times, 50 times, or 100 times as a JPEG. And then if you blow it up and look at the pixels closely, you can see that they're changing with the progressive saves. So, I mean, I don't think you're going to be sitting at your computer hitting Control-S 100 times, but think about how many times you're saving an image when you share it or transfer it with colleagues. Now, each time you're saving that image as a JPEG, you are degrading your data. So, talk to you now about experimentation and data analysis collection. Now, I'm just going to spend some time talking to you about preparing your manuscript. Remember, both the text and the figures need to be presented transparently so that reviewers can assess your work. And if your paper is accepted, readers can understand and replicate your results. These are just some questions I think you should be asking yourself as you're preparing your manuscript. Regarding the text, are you describing your results accurately? Can somebody replicate your experiment with the protocols you have written? Have you listed all your reagents and materials? And do you have a data availability statement? Regarding your figures, are the figures an accurate representation of your results? That means, do they look like the original capture of your data? Did you acquire your data in the linear range? And do your images show the appropriate context? So I'm going to start off by talking a little bit about your text. So in this example, what the authors have done is they've either taken a wild-type mouse or a BDL knockout mouse and taken a bone marrow suspension from them, and then plated them in soft agar cultures in the presence or absence of cytokine. A week later, they go and count the number of colonies on the plate. So in C, what the authors decided to do was they decided to plate the results here in this table as just the mean plus or minus the standard error of the mean. And they didn't like how that was going to look for their figure or for their paper, so they decided to then instead start the y-axis off at 40 rather than 0. And then they put this into their figure, or into their paper, and then they go on to the text to say that the difference between the two was dramatic, indicating that this gene is absolutely required for proliferation of bone marrow cells. So there are actually a lot of issues with the way the authors have presented their data here. In addition to how they presented the graph, as well as their experimental design, they completely overstated their results. It would have been better for them to have said that BDL is needed for a full response. So overstating your results by trying to make things sound more significant than they actually are going to be a huge turnoff for any reviewer who reads your manuscript, and it's going to be a misrepresentation of your experimental results. So probably an often overlooked part of your text is your figure legends, but these are really important because these are what are going to guide your readers through your experiments. So remember, they should be able to stand on their own. That means if the reader has just your figure and your figure legends, they should be able to tell exactly what's going on in your figure with just those two things. The legend should also have a title that describes all the figure panels within that figure. Usually I've seen that written up as a statement of the results of the figure, but it can also be descriptive. It can just say the type of experiment that was performed. It should also provide a description of the techniques that were used. That means including the experimental model, the conditions that were tested, the controls that were used, the number of biological and technical replicates, and any statistical tests that were applied. And finally, you should define any features that were in the figure, such as symbols or colors or any non-standard abbreviations. So I am probably a little biased here, but I think the most important section you're going to write in your text are going to be the experimental procedures section. If it doesn't provide adequate detail, readers are not going to be able to tell if the studies were performed appropriately, nor are they going to be able to replicate your results. So these are the key elements of this section. First, it should describe the materials that were used in the study and how they were prepared. The protocol needs to be described with enough detail such that a qualified investigator would be able to repeat those experiments. That means explaining how any measurements were made or any calculations that were performed. And finally, you need to state any statistical tests that were used to analyze your data set. So providing enough detail of the materials used in the manuscripts means providing enough information that somebody can uniquely identify the materials that were used in the study. That means if you used antibodies, you should include sources, your dilutions, any catalog numbers. If you've used a cell line, you should include the source, whether or not they're authenticated, whether or not they're contaminated. If you've used animals, you should specify the source, the species, the strain, sex, age, and any relevant details of husbandry. You should also specify the genetic background if you used any transgenic animals. For chemicals and reagents, you should identify the source. And if you've prepared any of the reagents, you need to provide information regarding the solvent and the purity. So a lot of publishers are also recommending to their authors to use these research resource identifiers, or RRIDs. So these are persistent and unique identifiers for antibodies, organisms, cell lines, and tools. So what's really neat about these identifiers is that they're machine-readable ID numbers, so you can search across multiple, or actually all studies that have used a particular research resource. And because they are persistent, you can still identify a resource even if the company has changed the catalog number or they've discontinued the product. So regarding the protocol, when you are reading a paper, what is useful for you when you are reading the methods section? For example, saying the samples were centrifuged doesn't give you any special information such as the speed or the time or the temperature. And what really frustrated me when I was doing bench work was seeing these words, "The experiment was performed as previously described," and it would give a reference. Then I would go look up at that reference and would refer to another paper, and this could go back several times and was really annoying. So what I would recommend is just briefly restating the methods that were used in that experiment. That way readers aren't frustrated having to dig up exactly how that experiment was performed. If you are describing a new method that would benefit from like a really detailed protocol, like providing the type of tubes that were used and like how you twisted the tube a certain way to get your lysate to do something special, you should consider submitting it to either bioprotocol.org or protocols.io. So an added benefit to submitting a protocol to one of these sites is that readers can submit questions if they have any, they need any part of your protocol clarified at all. So the final thing I wanted to mention about your text are data availability statements. You should consider including them in your submissions. So these statements explain to readers how they can access any of the data that's found in your article. They can also improve the discoverability of your research by linking it with appropriate data sets. And you're going to see more and more publishers are going to be requiring this information in their submissions. Just remember when you're writing up your text, the more information that you include, the easier it is for readers to follow your thought process and to replicate your results. And most journals and publishers, they don't have a character limit anymore for their articles, so there's no reason to restrict the content that you're putting into your manuscript. So I've talked to you about writing up your text, but your figures are visually what readers are going to be using to assess the quality of your work and to make sure that the conclusions you've made in your paper can actually be drawn. So one of the biggest problems that we have when we're analyzing images is that the author has either prepared their figures or they've archived their data in PowerPoint. So I know PowerPoint is a really easy program to use and super convenient, but it's a program that's intended for on-screen presentations only and not for handling high-resolution graphics. So in this example here, I've resized an image in Illustrator, and here I've resized an image in PowerPoint. You can see there's a vast difference in the quality of the image here. So what PowerPoint does is it automatically downsamples all your images to 72 dpi, which is not suitable for publication. Now, once that image is downsampled, you're not going to be able to recreate a high-quality image from that. So if you really want to make a high-quality figure, you really need to be using a program that's intended for handling high-resolution images, such as Illustrator, Canvas, or Inkscape. So also when you're making your figures, you also need to be sure you're showing the appropriate context with your figures. So what does that mean? So for example, if you're showing any blot data, if you crop too closely around the bands of interest, it's not going to give your readers any information about the specificity of your probe. Also, if you show more area or more context around your bands of interest, a reader might notice something that you might have missed, like a cleavage product or a differentially modified form. And it's not just blot data. You should also be showing the appropriate context with cells, images, or cell images, organs, or organisms. You need to be showing scale bars so that readers can assess the relative sizes of whatever you're observing. So finally, I just want to go over some common traps that authors may find themselves in when they're preparing their manuscripts. So we see this quite often when we're analyzing images. These are placeholder images. So what will happen is that an author will start building their figures before they finish doing all their experiments. So what they'll do is they'll put a placeholder image in their figure so they have an idea of what that final figure layout's going to look like. And instead of removing that image later, it gets left in. So I am going to discourage this practice completely. It has the huge potential to backfire on you. If it's discovered during review, your manuscript could get declined. Also, there's no added benefit for constructing your figures this way. For me, it's a lot easier to tell what experiments still need to be done if I can see where the holes are in my figures. So something else that can get authors in some hot water is being reckless with adjusting or manipulating an image. So in this example, the authors felt that these other cells were really distracting, so they just decided to remove them from the frame. So I've also seen this with blot data. The authors wanted the blots to look much cleaner than they actually were, so they decided to fudge some of the background bands. Just remember, most journals now have adopted this policy that was originally developed by the Journal of Cell Biology. No specific feature within an image may be enhanced, obscured, moved, removed, or introduced. The groupings of images from different parts of the same gel fields or exposures must be made explicit. Any adjustments you make to brightness, contrast, or color balance are acceptable if they are applied to every pixel in the image, and as long as they do not obscure, eliminate, or misrepresent any information present in the original. This includes the background. Any nonlinear adjustments must be disclosed in the figure legend. So I realize that when you open up Photoshop, there are all these wonderful tools that you can use to beautify your image. Just remember, most of these are off limits. If you have to adjust an image for publication, you really only need to be using brightness, contrast, and levels. And even if you do use those, you need to make sure they're not misrepresenting your original image. Also, if you make any adjustments to an image, they need to treat each pixel equally, and you should only make adjustments to a copy and not the original. So the last point I wanted to mention about some common traps relates to a bias in the literature known as p-hacking. So this happens when a researcher tries out multiple different analyses and then selectively reports the one that's statistically significant. So in this cartoon, the authors wanted to see if jelly beans cause acne. So they go on and do their experiments, and they come back and realize, no, it didn't produce a statistically significant result. So then they go look and reanalyze their data set multiple different ways. And they finally find one color that produces a statistically significant result, and that becomes news. So green jelly beans are now linked to acne. So don't eat any green jelly beans. So some practices or behaviors that can lead to p-hacking are include conducting an analysis before finishing your data collection to decide whether or not to continue, recording many variables and deciding which ones to report after analysis, deciding whether or not to include or exclude outliers post-analysis, or stopping the analysis once you've reached a significant p-value. Now, you may not even realize that it's happening, but there are some things that you can do to avoid this bias. First, you should carefully design your experiment and carry out that study and analysis according to that plan. You should also completely describe how the analysis was performed so that readers can evaluate whether or not you performed the correct one. So one researcher, in fact, was heavily scrutinized in 2017 when he wrote a blog post admitting to questionable research practices. So Brian Wansink is currently the director of Cornell's Food and Brand Lab. So in the blog post, Wansink stated that he encouraged a graduate student to reanalyze a data set multiple different ways, which called into question whether or not the results have been p-hacked. So this caused a lot of outrage, and many of his publications were investigated. Some of the issues that were found in his papers was the misreporting of research data, problematic statistical techniques, and a failure to properly document and preserve research results. So he's up to about 17 retractions right now, and he will be stepping down from Cornell later this year. So the example I just gave you of Brian Wansink segues into the last portion of my talk, which is any consequences that may occur. So I spent some time talking to you about some potential rabbit holes that authors may go down when they're performing their experiments, analyzing their results, or writing them up. But I also want you to keep in mind there are real consequences for any of the issues I just mentioned. So these consequences can affect the scientific enterprise as a whole, but they can also affect you individually. So in the past decade or so, irreproducible research, which is a result of detrimental research practices, has really taken center stage in the general discussion about science. There are estimates that about 75 to 90% of all findings in preclinical research presented in high-profile journals are not reproducible, which can decrease public confidence in the scientific enterprise. So eroding public trust in science can have an impact on future funding. Our livelihood is predicated on public trust and the belief that the questions that science wants to address are for the greater good. So we, as in researchers, institutions, and publishers need to ensure compliance with good scientific methods and transparency in data reporting to ensure public trust in our endeavors. It's also our responsibility to ensure that we're not unnecessarily wasting resources, because this is also going to feed back into public trust. And there's just not a monetary consequence. The outcomes of research reports are being used in the clinics, so there are lives at stake when the research is flawed. So Paolo Macchiarini was a superstar at the Karolinska Institute, where he led the Advanced Center of Translational and Regenerative Medicine. So he rose to fame when he transplanted artificial tracheas that had been seeded with patient stem cells to prevent organ transplant, or to prevent organ rejection. So these were hailed as a big breakthrough at the time, but eventually came to light that out of the 20 patients who received these transplants, only 3 survived. So what's even more shocking are some of the details that emerged after all this happened. Depending on which CV you looked at, Macchiarini had either performed a fellowship at UAB or earned his master's there before he moved to France to get his PhD. He also listed being a full professor at 2 different institutions, one in France and one in Germany, when in fact, none of this was true. He was also engaged to a producer from NBC, and he had convinced her that the Pope was going to officiate their wedding ceremony at a summer palace, and that the guests were going to include Obama, Putin, and the Clintons, when in fact he was still married to his wife of 30 years. Macchiarini was eventually fired, and the center was closed, but the damage had already been done to these patients. And finally, what's probably most important to you guys, there could be possible personal consequences to you. Your reputation may suffer. Your paper could be corrected or retracted or discussed online on forums such as PubPeer. You could be sanctioned from your institution or the Office of Research Integrity. Your research could be scrutinized in the future by the National Institutes of Health. You could lose your medical license. You could lose your job. Your PhD could even be revoked. You could even serve jail time. So all of these actions are going to take up a lot of time, money, and resources, are going to impact you and those around you negatively. So I would strongly urge you all just to take the time now and make sure you're performing everything as carefully and methodically as possible. So I've shown you all today some places where researchers can make some missteps in the spectrum of consequences. Just want to close by bringing you back to one of my starting slides showing the research life cycle. There are reasons why institutions, funders, and publishers have policies and best practices in place. They're really a protection for you, as well as the subjects of your research, and the funds that enable you to do that research. At each one of these steps here, there should be a set of checks on integrity, such as there should be a set of checks on integrity, transparency, and reproducibility. So during this phase, grants are funded based on strength and merit. During this phase, if you're a first author on a paper, you're likely discussing your results with your lab mates as well as collaborators, and you should be getting feedback. Now, if you're a co-author on a paper, you should also have access to any of that data that's in your manuscript. Remember, you're going to put your name on that paper, and you want to be sure you can stand behind the results of that paper, because you're going to rise, and you could also potentially fall with that paper. When you come to this phase, when you submit your article to a journal, it should undergo a rigorous peer review process. And finally, at this phase, once your paper is published, your paper is out there for critique by the scientific community. So, if we want to keep this cycle going, we want to make sure that everybody is doing their part in performing, reporting, and evaluating transparent and reproducible research. So with that, I'd like to thank you all for your attention. If there are any questions that you may have that don't get addressed in this Q&A session, please feel free to contact me at my email address. And I also want to direct you to 2 resources that are available from ASB&B and the JBC. And with that, I'd be happy to take any questions.